Thank you for tuning in. This is Lance, your host of Strange Bites, where we take real science and wrap it up in a creative story and serve it in 15 minutes or less. If you're enjoying the show, please hit that subscribe button on your favorite podcast app. Give me a follow on social media. And now, let's jump into the next bite. You are in a dimly lit laboratory in Minnesota. It's a quiet night. Incubators hum like sleeping beasts. The air carries the faint metallic tang of chemicals. On a bench sits an ordinary glass flask filled with a murky, swirling soup. Inside that soup, something impossible is happening. Scientists have built a cell from scratch. They started with purified chemicals, nothing that was ever part of a living thing, and assembled something that eats, grows, copies its own genetic code, divides into daughters, and even competes with its neighbors for food. They nicknamed it Spudcell because under the microscope it looks like a lumpy green potato, and because it echoes Sputnik, the satellite that launched the space age. This little chemical blob is the closest humanity has ever come to creating life-like behavior from pure non-life in a lab. This is episode 42, a deep, weird, wonderful bite into one of the wildest discoveries of 2026. The story of Spudcell and what it means when humans learn to build life from the ground up. So come on, let's dive in. Our story begins with a question that has haunted scientists and philosophers for centuries. Can chemistry alone give rise to something that behaves like life? For decades, researchers tried two main roads. One was top-down. Take a simple living cell and delete genes until you had the bare minimum. That gave us impressive minimal bacteria, but they still carried the mysterious baggage of billions of years of evolution. The other road was bottom-up. Start with dead chemicals and try to spark life-like behavior. People made membranes. People made tiny factories that could read DNA and spit out proteins. But getting all the pieces to work together in one self-sustaining system, that was the holy grail no one had fully claimed. Until now. In the Adamala lab at the University of Minnesota, led by synthetic biologist Kate Adamala, the team decided to try something radically clean. They would build everything from known purified ingredients so they could understand every single piece. No black boxes, no, we don't know what this does. A fully chemically defined system. First, they needed a boundary, something to keep the insides in and the chaos of the outside world out. They used liposomes, tiny hollow spheres made of the same fatty molecules that form real cell membranes. When you mix certain lipids with water, they spontaneously form these bubbles. Nature does it. The scientists just gave it the right recipe. These became the skin of the Spudcell. Inside that bubble, they placed the protein-making machinery. Not a sloppy extract squeezed from real bacteria, but a precise, defined mixture called the pure system. Protein synthesis using recombinant elements. It contains exactly 36 purified enzymes and ribosomes. Think of it as a clean, minimalist factory. Feed it DNA instructions and it reads the code and builds the proteins the cell needs. No mystery ingredients. Every molecule is accounted for. Then came the genome, the instruction manual. They designed a custom set of DNA roughly 90,000 base pairs long, split across 7 to 9 separate small circles called plasmids. That's tiny compared to a human genome, 3 billion letters, or even a typical bacterium. Most of the genes came from the common gut bacteria E. coli, and from viruses that infect bacteria, with one glowing gene borrowed from a jellyfish so the cells would light up under a microscope and be easy to watch. This genome encodes the core behaviors, how to make proteins, how to copy DNA, how to feed, and how to divide. Now the real magic begins. The spud cells wake up in their flask. They read their DNA and start producing a special protein called alpha hemolysin. This protein inserts itself in the fatty membrane and displays a little chemical tag on the outside, like a key or a fishing hook. Floating in the same soup are smaller feeder liposomes packed with fresh supplies, more lipids for membrane growth, enzymes, ribosomes, small molecules, even ATP, the energy currency of life. When a spud cell's hook finds a matching tag on a feeder liposome, the two bubbles fuse. The cell literally swallows its snack. It grows larger as it incorporates the new membrane material. It's genetically controlled feeding. The DNA decides how much hook protein to make, which decides how fast the cell can eat and grow. No complex metabolism yet. The cells are still outsourcing a lot of their kitchen work to those prepared snack packs. But the link between genes and behavior is there. As the cell grows, the DNA gets copied using enzymes borrowed from a bacterial virus. Then comes the hardest part in synthetic biology: division. Real cells use an elaborate internal skeleton, a cytoskeleton, and dozens of coordinated proteins to pinch themselves into two neatly parceled out DNA. Building that from scratch has been a nightmare for years. Spudcell cheats beautifully. It doesn't bother with a skeleton. As proteins pile up on the inner surface of the membrane, they create mechanical stress and crowding. The membrane buckles inward under tension and splits. Scientists can add a little extra help, special linker proteins, to make the pinch cleaner and more reliable. The two daughter cells inherit copies of the genome and float off to start their own cycles. It's crude, it's mechanical, and it works. Now here's where the story turns competitive and strangely evolutionary. The team created a mutant version of Spudcell with a stronger promoter, basically a volume knob, for the gene that makes the fusion hook protein. These mutants make more hooks, grab more feeders, eat faster, grow faster, and divide faster. The researchers mixed equal numbers of normal and mutated cells in flask with limited snacks. After just 5 generations, the mutants often took over, sometimes climbing to 60% or more of the population. When food was made even scarcer, the advantage grew stronger. The faster feeders literally outproduced their neighbors. It's not full Darwinian evolution yet. The mutation was deliberately introduced by humans, and the cells don't generate new random mutations on their own in these experiments. But it is selection and competition operating in a completely synthetic, chemically defined system. Genotype influences phenotype, which influences reproductive success. In a flask, in a lab, from pure chemistry. Of course, Spudcell is still a fragile prototype, what Automala herself has called an incredibly wimpy organism. It needs constant outside help. The ribosomes it carries eventually degrade, and after about 5 to 10 generations, the system runs out of steam. Genome distribution during division isn't perfect yet. Only about 30% of the daughters get the full set after several rounds. The cells only function under tightly controlled lab conditions with regular feedings. They're more like sophisticated, high-maintenance chemical robots than independent life forms. They won't be escaping the lab anytime soon. Still, this is monumental. It's the first time a fully bottom-up, chemically defined system has completed an entire basic cell cycle, feeding, growing, replicating its genome, dividing, and showing heritable advantages across generations, using only known purified components. A 190-page reprint detailed every step. The work was even shopped around journals. One reportedly said it wasn't real biology. So the team went open with it via the new nonprofit Biotic, co-founded with synthetic biology pioneer Drew Indy and others. They want to make Spudcell a shared open source chassis that the global community can improve together, like Linux for building life. Now we reach the part that lingers long after the story ends. What does it actually mean when humans can assemble something that checks so many boxes for life from a shopping list of chemicals? First, it forces us to confront our definitions. Life has always felt special, almost magical. that elusive spark separating the living from the non-living. Spudcell suggests life might be more like an emergent property of chemistry under the right conditions. Compartmentalization, information storage and copying, energy use, even if outsourced, growth, reproduction, and the capacity for heritable variation that leads to selection. There's no single line you cross. As Adamala has said, life isn't binary. This little potato-shaped blob sits right on the blurry edge. It also gives us a powerful new tool for understanding how life might have begun on early Earth, or how it could begin elsewhere. If we can coax these behaviors out of purified molecules in a flask, perhaps the first protocells arose through similar self-organizing chemistry in some ancient pond or hydrothermal vent, without needing a fully formed modern cell to start with. If we look ahead, the implications get even stranger and more exciting. Synthetic cells like this could become programmable microscopic factories. Imagine custom-designed versions that produce medicines on demand, break down pollutants, create sustainable biofuels or novel materials without toxic petrochemicals, or even act as smart drug delivery systems inside the body. Because everything is chemically defined and modular, those separate plasmids are like swappable apps. Scientists could, in theory, mix and match functions with far more precision and safety than hacking natural organisms. The open approach via biotic aims to accelerate this while keeping safety and ethics front and center. But power always brings shadows. As these systems get better, able to make their own ribosomes, sustain themselves longer, generate their own variation, perhaps even evolve more independently, we step into new ethical territory. Who gets to decide what kinds of synthetic life we bring into existence? What safeguards prevent unintended consequences if something more robust ever left the lab? Could we accidentally or intentionally create organisms that compete with or disrupt natural ecosystems? And on a deeper level, if life is something we can engineer from a parts list, does that cheapen our sense of wonder about the natural world? Or does it amplify it by showing us how extraordinary the chemistry of existence really is? There's a profound humility here too. Spudcell is still wimpy. It needs constant babysitting. It reminds us that even our most impressive creations are fragile and dependent. At the same time, it shows we are becoming creators at the most fundamental level, not just reshaping existing life, but assembling new instances of lifelike behavior from the ground up. That's a responsibility that should make us pause. Spudcell isn't just a scientific milestone, it's a mirror. It reflects our curiosity, our ingenuity, and our drive to understand the universe by rebuilding its pieces. It also reflects the question we can no longer avoid. What kind of stewards will we be of this new power? Will we use it to heal, to sustain, to explore responsibly? Or will hubris lead us somewhere darker? As the last Spudcell daughter splits in its flask and the glow fades under the microscope, one thing is clear. The line between chemistry and life is thinner and more wondrous than we ever imagined. That's it for today's bite. I hope you enjoyed the story. If you did, hit that subscribe button on your favorite podcast app. Give me a follow on social media. Until next time, stay strange and question everything.